Welcome to AP Biology, Unit 4.1 to 4.5 Review, Cell Communication, Cell Signal Transduction, and Feedback Loops. This is Dr. Joseph. In this review, we are going to look at cell-to-cell communication. We're going to look at what ligands are and what receptors are, and how the process of signal transduction so how the process of communication involves reception, transduction, and response. And we are going to look at the different types of cell communication and focus on three kinds of receptors. And as we discuss them, we will understand the roles of kinases and what signal cascades are, and how they amplify signals and why they are critical for varying response and control. Finally, we'll end by looking at homeostasis and how negative feedback and positive feedback loops contribute to homeostasis, and finally, how changes in signal transduction pathways can help modulate them. So, all cells need to communicate with each other, whether they are single cells or whether they are multicellular organisms. The cells need to connect and communicate with each other because they need to be in touch with the external environment, and they need to be able to sense it and respond to it. Response is a characteristic of life. So, when cells receive the signal from the outside, they can respond by changing their gene expression, how they are growing and dividing, and how they contribute to the response itself. By releasing hormones so that other cells around them are impacted, and so on. So, all signal transduction processes start with a message, and that message are usually molecules, small molecules, or proteins, or lipids. So they are called ligands. So is a general word that talks about molecules that help carry a message, and that bind to a receptor. There are two types of ligands. One made of proteins, the other one is made of lipids. As we know from our previous understanding of the biochemical characteristics of lipids and proteins, lipids are uh, non-polar molecules, which means they are not charged, and therefore they can pass freely across cell membranes, because cell membranes are have a the core of of a cell membrane is essentially lipids, fats, so it is non-polar. So lipid ligands can freely pass through cell membranes and, if needed, nuclear membranes. Whereas protein ligands are water soluble, they are polar, and they are unable to traverse the cell membrane. So, in that case, what they have to do is they have to bind to receptor proteins that are found on the membranes of the cells. So, therefore, you end up with two kinds of receptors that are based inside the cell, so that they can interact with lipid based ligands, and these are called intracellular receptors, inside the cell receptors. And those that are meant to receive the protein ligands, so they must be embedded in the plasma membrane, and they are called as plasma membrane receptors or cell membrane receptors. And when you look at a plasma membrane receptors, usually it has three parts. It's got a portion that is sticking outside the cell membrane, away from the cell. That's called the extracellular domain. Domain means region. And then you have a region which is bound to the plasma membrane, which is filled with hydrophobic amino acids. And then you have a portion which is sticking inside the cell membrane towards the cell, and that is the intracellular domain. So this will become important when we study G protein receptors and tyrosine kinase receptors and so on. So we'll come to that in a second. So signal transduction involves three steps. It involves receiving the signal, which is basically receiving the ligand, transducing the signal, which is basically carrying that signal from the place where it is received to the place where it is going to elicit a response, and then the response. That is the third step. So here are some examples. Lipid-based ligands, um, you have modifications of cholesterol, that includes steroids like testosterone, estrogen, progesterone, and as we discussed, they are able to pass through the plasma membrane because they are non-polar. So you have a steroid hormone and you have an intracellular receptor. The steroid hormone passes through the plasma membrane because of its non-polar nature, comes and binds to it, and now together this can go and create a response. In this case, we are showing a DNA, and DNA needs to be transcribed into RNA, and that RNA has to be translated into protein. We are going to study transcription and translation in much detail later on, but what we have here is that there must be a signal that should bind to the DNA that says go ahead and make your RNA or don't go ahead and make your RNA. So these are called as transcription factors. So binding of the steroid hormone to the receptor protein creates a transcription factor in this case, which allows it to bind to the DNA, therefore acting as a transcription factor, causing the production of messenger RNA, which then can be converted into protein. So this is lipid-based ligands with an intracellular receptor. Then you have protein-based ligands. Examples are insulin, epinephrine, which is adrenaline. There's a whole bunch of them, and because of their polar nature they cannot pass through membranes. And here you have a receptor protein, which is an intracellular cell membrane receptor, plasma membrane receptor. So the hormone in this example, which is water soluble, comes and binds to this receptor protein and therefore elicits a cascade of events, which is a series of events. Think of the domino effect. One thing leads to another. So there is a series of events that could happen that ultimately results in a response. So in both of these cases, just as a terminology, the cell in which all of these responses are being created are called as target cells. Okay, let's look at the three steps: reception, transduction, and response. Reception is when a ligand binds to a specific receptor. Not all cells have receptors for every ligand out there. So this is contributing to the specificity. Only even though the hormones or any other ligand is going to be present all over the place, only those that have the receptors for that particular hormone or that particular ligand are going to respond to it. Once the receptor and the ligand are bound to each other, there is a shape change in the receptor, and that's a common thing we see when something binds to a portion of a protein. That protein can undergo conformational change. We saw this when we were studying substrate enzyme interactions. So this change initiates the next step in the process. So here again we are going to look at this in more detail. So here we have a hormone, epinephrine, which comes and binds to a receptor, and that changes the shape. That activates a G protein, which goes and activates an enzyme that goes ahead and activates a second messenger, which is a small molecule that's going to carry information and transmit the information, and that activates a series of other proteins that essentially creates a modification of an enzyme, or it could go ahead and change gene expressions. Which is, when you say gene expression, we are talking about which proteins are being made and which proteins are not being made, or which RNAs are being made and which RNAs are not being made. That's what gene expression is. So after that process happens, the series of events, after that binding event, reception happened, the whole series of events that I described, where the G protein gets activated and the adenylyl cyclase and then the second messenger and the series of kinases, all of these things is called as signal transduction. It is a series of chemical reactions triggered by the ligand binding to the receptor that helps the cell respond. So this is usually very complex, and you'll see in a picture later how complex some of these events can get, because it's not always one leading to the other, there could be crosstalk between different signaling pathways at any given time, each of them influencing the other. So it's a very, very complex bunch of reactions. And a lot of research is still being carried on to understand how these signal transduction pathways work and how we can interfere with those signal transduction pathways so we can change the response, modify the response. So cascades are a series of reactions. One molecule activates a multiple group of molecules and therefore amplifying the signal. This process is called as signal amplification. So one binding here can activate multiple second messengers, and multiple second messengers, each one of them can go ahead and activate multiple protein kinases, and each one one of those can amplify the signal further, and so on. So one of the key types of proteins that we see in the signal cascade pathways are kinases. Kinases are enzymes that can transfer phosphate groups to other molecules. Remember that addition of phosphate is a big strategy that cells use for changing response. So when phosphate molecules, that is PO4 molecules, bind to proteins, they can change the activity of the proteins. They can change the shape of the proteins and therefore their function. And but it cannot be a one-way process. Once you have something phosphorylated, and kinase belongs to a general category of enzymes called phosphorylases, those that add phosphates. Now, once a phosphorylation even happens, it cannot be one way. We need to replenish the non-phosphorylated versions of the proteins. So therefore, we have enzymes called phosphatases that remove the phosphate groups so that they can be inactivated. So phosphorylases such as kinases and phosphatases work hand in hand so that they can create a cycle of events that can continue activation signals. And a part of this is secondary messengers. Um, Here, one of the most common secondary messengers is cyclic AMP. So, for just to refresh our thoughts, if you remember, ATP, adenosine triphosphate, we know is the energy currency of the cell. If you remove one phosphate from that, it becomes adenosine diphosphate or ADP. You remove two phosphates and you just leave it with one phosphate. So that is called as adenosine monophosphate, AMP. And the cyclic version of that, chemical version of that, is called as a cyclic AMP. And cyclic AMP can act as a messenger. It can carry message from one part of the cycle to the other part of the cycle. So second messengers are non-protein molecules that play a critical role in signal transduction. So after that signal transduction process has happened, the ligand binding has created enough molecules to elicit a response. So in some cases, it is about turning on the genes, which is basically transcription modification. And as in this example of epinephrine, one of the responses, there can be many responses. One of the responses is it changes the gene expression of a protein called CREB, which can help break down glycogen into glucose. So moving on, let's look at the types of cell communication, types of signaling. You can broadly categorize it into three major categories. We usually categorize signal transduction based on the distance between the cells that are signaling to each other and the strength of the signals. So if you are looking at distance, you can say cell communication could happen by one cell juxtaposing, which is touching another cell. So that kind of signaling is called as you see this a lot in immune cell responses. Immune cells can bind to other immune cells. One cell will express an antigen, which is a glycoprotein or a glycolipid molecule on its surface, and the other cell is going to recognize that and bind to it, and the binding is going to create a series of responses. So that's an example of direct contact signaling, otherwise called just juxtacrine signaling. Another type of direct signaling is using gap junctions in animal cells. So, gap junctions are, you can think of them as pipes that are connecting the cytoplasm of two cells, where materials from one cell, information and signals from one cell is transferred or moved over to the next cell. And therefore, they are able to create consolidated responses. And in plants, though, you have an additional barrier, which is the cell wall. So the cell wall in plants have these holes, these tiny holes, through which the connection, the plasma of cytoplasm of adjacent cells can be connected through pipes. So these tubes are called as, these holes are called as plasmodesmata. So that is all direct contact signaling. The other form of signaling is local signaling, and local signaling, one of the major things here is paracrine signaling. Paracrine signaling is a cell produces signal so that the nearby cells, the target cells, which are in the near vicinity, are activated by the response. They have ligands for that they have receptors for those ligands, and that are created nearby, and they respond to it. Um, and there are a couple of specialized forms of paracrine signaling. One is autocrine signaling. Autocrine signaling is where a cell produces a signal, a ligand, and it has receptors. That cell itself, the cell that produced the signal, has ligands for the signal that it produced. So in other words, as soon as it produces its ligand, that ligand can come and bind to itself and further activate the signaling pathway. So that is called as autocrine signaling, or it can even act as an inhibitor. What it produces at some point can be used as a control knob, so to speak. So auto means self. Autocrine signaling is when a cell produces signals that can bind to its own receptors. And another form of paracrine signaling, or another direct signaling pathway, is neuronal signaling. So neurons, as you know, are nerve cells, and which conduct action potential charge across them. And neurons have these long arms called axons, and axons can communicate with other axons or with target cells such as muscle cells, um, in a very interesting way. So between the neuron, let's look at two neurons side by side. They have a small region, a space that separates them, so they are not in direct contact. And in that space, you have one cell secreting some chemicals called as neurotransmitters, which are received by receptors on the target cell, and that in turn helps create a response. So paracrine signal, local signaling includes paracrine signaling, where one cell signals to nearby other cells, or specialized versions of that where there's autocrine signaling, a cell can signal itself, and neuronal signaling, which is called as synaptic signaling. And then we have endocrine signaling. Endocrine signaling is hormonal signaling, where we have cells produce ligands, and those ligands can be transported through the body by blood, and it creates a response and at a location distant from itself. So the brain can produce hormones, and that can influence how, for example, your digestion responses are created. So it's all kinds of long distance signaling using hormones. So that is endocrine signaling. So we're going to look at each one of them in a little bit more detail. Direct contact signaling, the inducer or the ligand is not coming out of the cell. So either it is transferred from one cell to another cell through gap junctions, which we just saw, or plasmodesmata, or by juxtaposing, where one cell juxtaposes or comes into close contact with the other one, as we see in immune cells. This is just a close-up version of that. You can see molecules being transported from one cell to the other using these tubes. These tubes are made of these proteins called connexins. Connexins form, a tube here, it forms a tube here, and those two tubes attach, and it's an engineering marvel there. And then you have cell recognition and immune responses. And in paracrine signaling, cells have to be close to each other. It helps coordinate local coordination of activities with the neighbors. Um, and it's particularly important in development. When you have one embryonic cell becoming the plethora of human cells. A human body has about 200 different types of cells, and so all of those differentiation has to be very, very closely coordinated. So a cell has to say, I am becoming this, and this is what I'm and just to know that this is what I'm becoming, I'm sending out these signals near me, and the ones that receive that, I say, okay, that role is already taken up, I'm going to become something else. And not exactly in this kind of conversation, but you see where I'm going with that. So it's important in development. Autocrine signaling, as we just saw, includes, essentially means ligands binding to receptors on the own surface, and it's important in reinforcement and in the overproduction of growth factors in cancer progression, and many times it is. I shouldn't say many times. Sometimes it is, when a cell secretes a ligand, it can be paracrine signaling as well as autocrine signaling. Examples of those are cytokines, which are part of immune responses. So the cytokines can bind to the cells that are near them, and also they can bind to themselves and reinforce that immune response. Then we have synaptic signaling, which we were just talking about. So here we have the synaptic cleft, which is the space between two neurons. Okay, so we have this tiny space about 40 nanometers long or 40 nanometers broad between the synapse of between the end of the axon of one cell and the other. So this neuron is called as the presynaptic neuron, the one that is sending the signal. The one that is receiving the signal is called as postsynaptic neuron. And so when the electrical signal comes here, there are vesicles that contain these molecules called neurotransmitters. And these neurotransmitters, once they are activated by the signal, can go ahead and release into the synaptic cleft. And the postsynaptic neuron has these neurons on its surface which can receive that ligand, the neurotransmitters, and in the process get activated. When it gets activated, it can create a series of responses. As we'll see later, with ligand-gated ion channels, this binding event can create the receptors to act like channels, to open up, and therefore allowing ions to come through, and therefore creating a voltage gradient in the postsynaptic cell, and therefore leading to a continuation of the neuronal response. And so the neurotransmitters can be it cannot be a permanent signal. Once it comes in, it has to be very quickly taken back in to the presynaptic cell. That's called reuptake, and or it can be degraded. So that's why you don't have a continuous flow of electricity. It happens in pulses. So there are three major types of so that's the part about the three different types. Let's talk about endocrine signaling for a quick minute here. So it involves movement of ligands through the cell, and even though it has to travel long distances, you can see in a coming up example that endocrine signaling can be extremely fast. Okay, so now that we have looked at the different types of cell signaling, we are going to look at the types of receptors. There are three major types of receptors. G protein coupled receptors. If it uses a G protein, to act as an on-off switch. Um, and then we have tyrosine receptor kinases, TRKs, sometimes called RTKs, but TRKs, truck, think of it as truck. Okay, tyrosine receptor kinases, they can add phosphates to tyrosines and create multiple transduction pathways at once. And this is really significant in cancer. And then we have, just as in the synaptic example we saw, the synapse synaptic interactions. We have ligand gated ion channel receptors, which act as gates that are controlled by ligands. So let's look at each one of them. G protein coupled receptors. We want to focus on this a little bit because it kind of helps us understand the actual anatomy of a signaling pathway or signaling cascade. So here we have a G protein coupled receptor, GPCR. It has a domain which is a portion that is sticking out, as we mentioned before. That's called as the extracellular domain. Then we have the transmembrane region, which is highly hydrophobic. And because, again, remember, all of these lipid tails are hydrophobic, so it has to be hydrophobic if it has to stay in this region. And then you have your intracellular domain, which is facing towards the inside of the cell. So here, in its normal form, in its inactive form, you have the G protein, which is a protein made of 3 subunits. Okay, it's called a heterotrimeric protein. Means 3 different types of subunits, heterotrimeric protein. In its inactive form, it is bound to GDP. Once again, just to remember, just like we have ATP and GDP, we have, ATP and ADP, we have GTP, guanosine triphosphate, which is, if you remember, DNA is made of 4 nucleotide, right? A, T, G, and C, adenosine, guanine, guanosine, tyrosine, and tyrosine, and I'm sorry, I'm messing up here. A, T, G, and C, and we have GDP, which is guanosine diphosphate, bound to the alpha subunit. Now, this is the inactive form. When you have a signaling molecule that comes and binds to this G protein coupled receptor, the GDP gets replaced by GTP. This is the place where it gets activated. When GTP replaces GDP, the alpha subunit separates out from the gamma and the beta subunits. And it now crawls along the inside of the membrane until it comes to a target molecule and creates a series of responses. Now, in epinephrine pathway, this target for the activated G protein is a molecule is an enzyme called adenylyl cyclase. Okay, so adenylyl cyclase, what it does is, when you have the alpha portion of the G protein bound to the GTP coming and binding to it, it gets activated, and then that converts ATP into cyclic AMP, and that creates a series of responses, which we'll see soon. Okay, so that is G protein coupled receptor, and then we have tyrosine receptor kinases. Before we talk about that, I want to talk about kinases in general. Okay, so kinases are, as we saw, phosphorylases. Basically, they add phosphates to other proteins and thereby activating them. And this creates a cascade of responses inside the cell, which is the signal cascade. So, in epinephrine adrenaline signaling, you have the G protein binding to adenylyl cyclase. Adenylyl cyclase converts ATP into CAMP, and CAMP activates a protein kinase. Okay, and when when a kinase is activated, it means it can go ahead and add a phosphate to another protein. So this kinase can go ahead and activate another kinase by adding a phosphate group to it. And now this phosphorylated kinase, in its active form, can go ahead and activate another kinase. So this keeps going on and on and on. And one of the responses for adrenaline is the production of glucose from glycogen. Remember that glycogen is a stored form of glucose, and when you're facing a fear or when a fearful experience, flight or fright situation, a flight or fight situation, you see a tiger on the road, or if you're encountering a very a car running towards you. So what happens is your muscles need to move really really fast so that you can survive. And so what happens is your adrenaline is secreted uh, by the adrenal glands, and which is in response to signals coming from the brain. And the adrenaline, otherwise called epinephrine, is going to activate the cyclic AMP through adenylyl cyclase, which is going to phosphorylate a bunch of kinases one after the other. And ultimately, one of the responses is phosphorylating an enzyme called glycogen phosphorylase, and glycogen phosphorylase can convert glycogen into glucose. Thereby, when glucose is released, it can be processed by the mitochondria to reduce to release ATP, and this ATP can be used for running away from the situation, escaping the situation, or standing back and fighting in that moment. Okay, so if you, I would urge you to spend a few minutes just watching this and understanding the series of events that happen. But one of the key questions is, why does it have to go through so many steps? Why doesn't something binding to a receptor immediately activate glycogen phosphorylase, and therefore convert glycogen into glucose? Now, here's the thing. One of the major reasons is the need for signal amplification. So you have one molecule of epinephrine um, binding to a G protein coupled receptor and creating a response. Um, and that response is when we each binding can activate about 100 molecules, 100 G proteins, and that in turn activates uh, adenylyl cyclase, um, and that adenylyl cyclase, each adenylyl cyclase can activate more, so you end up with about 10 power 4 molecules here of this first step, and that goes and activates protein kinases. Each of those protein kinases can go ahead and activate more protein kinases, different kinds of protein kinases, and that keeps going on. As you can see, this number keeps increasing, and when you are looking at conversion of glycogen to glucose, you have activated about 10 power 8 molecules with just the binding of a single receptor. The other advantage here, so amplification is one big part of the need for a signaling cascade. The other need for a signaling cascade is is the fact that you need to have control over these different steps. So instead of just having that one switch of either the ligand binding to the receptor or not, here you have all of these different steps which you can use for fine tuning the response. So speed and amplification and regulation or control, all of these factors come into play in making the signal cascading an important part of the signal transduction response. So here's just a quick, saying that phosphorylase kinases and phosphatases work in a cyclical way, so that they can keep the number of active and inactive enzymes, going, so the reaction can continue. Okay, now let's talk about the second kind of receptors, which is the tyrosine receptor kinases. So tyrosine receptor kinases um, have help attach phosphates to tyrosines, um, and thereby triggering multiple pathways at the same time. This is one of the key differences between G protein coupled receptors. G protein coupled receptors, as we saw, is one pathway here. This one creating a series of steps in one direction. Now, here you have multiple tyrosine binding sites, and depending on where which tyrosine is phosphorylated, you can create a different series of responses, a different set of responses altogether. Um, Again, that is a form of maintaining regulation or control over cell over signaling pathways. But the other difference is receptor tyrosine kinases are present as subunits, two subunits next to each other. And when a ligand comes and binds to it, an example of a ligand that uses tyrosine receptor kinase is insulin. So when a receptor comes and binds to these ligand binding sites, there is a dimerization process, meaning these two subunits come together to create one unit with two parts, dimer. And now the activated version, uh, the different tyrosines can phosphorylate depending on what ligand is binding, and based on that you can activate different signaling pathways. So here, uh, proteins are helpful in carrying the signal. So they have these inactive relay proteins. These proteins are called as relay proteins. So these relay proteins, basically saying they're relaying information, as in the game, right? So here the relay proteins which are inactive will come and bind to the phosphorylated tyrosine regions, and depending on which tyrosine was activated, was or phosphorylated, the relay proteins can bind to those, therefore creating a cellular response in one direction or a different kind of a cellular response. So multiple cellular responses can be activated. Once again, I would ask you to take a few minutes to look at this and completely grasp the series of events that are happening here. And the third type of receptors are ligand gated ion channels, and it's in the name. You just ligand gated ion channels. So it's a gate that is activated by the binding of a ligand, and that causes the entry of ions, which flow through the channel and facilitate a response. So ligand gated ion channels. Um, so ligands, for example, in the brain you have sodium and potassium and calcium. Um, so ions like sodium and calcium are not able to directly enter a cell membrane or pass through a cell membrane because they are positively charged. So they have to stay outside and outside the cell. But when you have a neurotransmitter binding to which is the ligand, neurotransmitter is a ligand here, it comes and binds to the ion channel, which is now covered with the gate. The gate opens, allowing the channel to pass or to allow the passage of calcium and sodium and so on. So inside the cell now you have an accumulation of Na plus and Ca plus. So the inside becomes more positively charged than the outside, and therefore you're creating a voltage gradient, which is now which now becomes part of the signal that is being transmitted through the neuron. So we saw how it's important to amplify the signal during signal transduction. The other part we also mentioned was that uh, the signal can cause a range of responses in different cells. So you have a simple example here. In response 1, you have a signal molecule binding to a receptor, creating a straight series of response, of signal transduction processes leading to a response. Here we have the binding of a signal molecule creating two possible responses. You have
a branching off, um, and so you can elicit two unique responses with the binding of that same signaling molecule. Or you have two pathways running side by side. You could have a G protein coupled receptor pathway, and you could have a tyrosine receptor kinase pathway, and the cell is very you know, frugal about what it tries to create in terms of proteins and things like that. So what it can do is it can use the same proteins. These different pathways can use the same proteins, and these proteins can be shared, or the usage of the one stops the usage of the other. So it kind of acts like an active crosstalk that regulates uh, the response. And um, then you have, for example, a different kind of um, receptor which can also receive the same ligand. So we have these same receptors here. You see this one, this one, this one. These are all the same receptors, but you have the same signaling molecule binding to a different kind of receptor, giving rise to a different kind of a response. So there is a lot of crosstalk, a lot of overlap, um, a lot of joint usage of resources between cellular signaling pathways. And you see how these responses are beautifully coordinated when we study apoptosis and cell cycle progression. Um, so apoptosis is regulated cell death or controlled cell suicide, where a cell that should not be existing anymore gets chopped up and placed in packages and then digested. So that way we are not releasing a cell with its toxicities into the nearby environment and causing a mass death of cells. So apoptosis is a very careful regulated cell death event, and it's a normal part of development. So here we have the formation of fingers. The beginning, you have your fingers as one big mass, and the tissue between those between the different fingers, what will become the fingers, starts undergoing apoptosis. So starts dying in a very carefully regulated way, creating a space between those digits. So um, the other thing I want to talk about here is maintenance of homeostasis. Homeostasis means um, the cell's effort to keep a stable internal environment even though the outside conditions are changing. So I'll repeat that. Homeostasis is the cell's effort to maintain a stable internal environment even though the outside conditions are changing. So it's important because your enzymes will work only within a narrow pH or a narrow set of temperature conditions. So you're required to keep your cell within that range. And so here we have, for example, a set point, which is the optimal point for the body, for the cell. Um, and when you have a receptor binding to the control center, a change may happen, and that change may elicit a response. And so the stimulus and the response, the stimulus creates a response, um, which may or may not take it away from a state of homeostasis. So based on that, you have two different types of pathways, because ultimately the cell's objective is to keep homeostasis, and so these two pathways are called either negative feedback or positive feedback. So negative feedback is the most common type of signaling. um, so in this case, the stimulus moves the cell away from its homeostasis, and the body responds by pushing the cell back into a state of homeostasis. Okay, so in other words, what your cell is doing is it's going against the direction of the stimulus. That is why it's called as negative feedback. So please don't get confused between the words negative feedback and positive feedback as being high or low, reducing or increasing. No, it's nothing to do with that. Is it going in the direction of the stimulus, or is it going against is the response going in the direction of the stimulus, or is the response going against the direction of the stimulus? So when the response is against the direction of the stimulus, that's when we call it as a negative feedback. So, for example, if your temperature in your house increases, you increase the air conditioning. So you turn down the heat, you increase the air conditioning so that it comes back to a normal stage. So that is negative feedback. Positive feedback is your stimulus is moved your cell from a state of non-homeostasis. Your cell is in a state of non-homeostasis, and your signal is pushing it away from that non-homeostatic state to a homeostatic state. Okay, so the stimulus is helping the cell already in a non-homeostatic state come to a homeostatic state, which is what it wants. And so your response to that kind of a stimulus is to amplify that stimulus, amplify that signal so that there is more of uh, a move to. Okay, here we are seeing an example of a negative feedback depending on how much blood sugar is there in the blood. If you have a lot of sugar, okay, that's not healthy for the cells because that creates a hypotonic situation for the red blood cells where they are going to um, uh, uh, so when you have too much glucose in the blood, then it means there is more water inside the red blood cells compared to the outside, which means water is going to flow out, which means that it's going to shrink. And so that's not a good situation. So what your body does, it responds by secreting insulin into the blood. Those are done by the beta cells of the pancreas. When the insulin is secreted out into the bloodstream, the liver takes up the glucose and allows the glycogen to be stored for long-term usage. On the other hand, when you have too little blood in your too little glucose in your blood, once again that is not an optimal situation. So what happens there is uh, the cell creates a hormone called cells create a hormone called glucagon, and the glucagon can stimulate the liver to produce um, enzymes to break down glycogen into glucose. So both insulin and glucagon are produced by the pancreas, but see how different the responses are. In both of those cases, the idea is to keep the blood glucose levels at a constant, and they do that by going against the direction of the stimulus. So that's an example of negative feedback. Example of positive feedback and is, like I said, we're starting with in a situation where there is a movement um, towards homeostasis by the stimulus, right? Because the system is already in a state of non-homeostasis. The stimulus is trying to get it into homeostatic state. A classic example is the birth of a baby. You have the baby's head pushing against the cervix, and the pushing event causes the stimulation of the pituitary gland to secrete oxytocin. Oxytocin is carried to the uterus, and oxytocin stimulates uterine contractions more. So the baby can push the cervix more. Again, that generates more um, um, oxytocin production, which causes more contractions and more pushing of the baby out. And this continues until until the baby comes out, and both the mother and the baby are now in a state of homeostasis because the baby is no longer stuck inside the mother and it has come out. That is the stage of homeostasis. That is a state of homeostasis. So that is an example of a positive feedback loop because the signal is reinforced by the response. Um, Another response, positive feedback response is this. When you have a fruit, a bunch of fruits, one of them ripens, what it does is it creates a gas called ethylene. That ethylene acts as a second messenger. It acts as a messenger, as a ligand, binding to the nearby fruits, and therefore telling them to ripen faster. So the ripening of one fruit in a group causes the ripening of other um, fruits in the nearby region. Okay, so that's it's amplifying that ripening response. So that's why it's a positive feedback loop. So since we were talking about apoptosis, I just wanted to quickly point out one little thing. This is just to reinforce the point that um, there are many different signaling pathways happening side by side at any given time, and there is a lot of overlap and connection and sharing, and that's happening between these different cycles and pathways. So, on one end, the signaling can lead to um, apoptotic response. On the other side, it can lead to um, unlimited, uncontrolled cell division, which is what cancer is about. So, somewhere in the middle is the balance, which is why all of this crosstalk and response to the outside environment becomes important in maintaining the uh, homeostatic balance of the cell. Just as we saw all of these different ways that eukaryotes connect with each other, single cells like prokaryote bacteria can also signal with each other, and they use a strategy called quorum sensing. So, quorum sensing is a population density dependent response to secreted chemicals. So, we have, for example, bacteria growing in a culture or on a plate. A single bacteria can produce a signal, but then that signal can be absorbed by the nearby cells, which is paracrine signaling, or it can be absorbed by itself, which would be autocrine signaling. Now, that alone is not sufficient for the cell to do enough uh, response, to create enough response. So you need a lot more signals to create a response. So the cell continues to continue to divide and keep sending out these signals. So as the new cells are formed, they create these ligands, which they put out into the medium again by paracrine and autocrine signaling. They are getting more and more activated, but nothing shows up yet. Okay, at this point, until there is so many cells producing so many of these ligands that there is enough um, concentration of ligands to activate a response. And so this event, as we just saw, is a density dependent situation for single cells to create a response. So that's quorum sensing, and it's critical for um, bacterial cell communication. Here's an example. So we have a few cells, signals, not much happening. When we have a sufficient number of cells, so that the cell signals are taken seriously, and then we have enough signals so that uh, the cells can actually step out of the biofilm or the colony that they are in and activate an infectious response. Finally, we are going to look at changes in signal transduction pathways. Um, Again, this is very intuitive. As long as you've understood the previous things, this is very intuitive. All we are saying is, if there is a mutation in either the receptors extracellular domain or intercellular domain or the transmembrane region, um, or if there are changes in the signaling molecule, which is the ligand, if there is a problem, or if there is a change in the many steps of the signal transduction, um, or the molecules that respond to it. In any of these scenarios, if there is a change, if there is a modification, then the signal transduction, it will be altered. It's again, it makes a lot of intuitive sense, but it's just a reinforcement there. So here as an example, we are looking at type 2 diabetes. We have insulin molecules coming and binding to a tyrosine receptor kinase, and that becomes a dimer, and that activates an enzyme, which activates another enzyme, and this enzyme can go ahead and cause the release of glucose transport vesicles, which are inside the cytoplasm. And when this change happens, when this modification happens, the glucose is now shifted. The glucose transport vesicles, transport proteins are shifted to the membrane, where they act as channels and allow the flow of glucose inside. But we have a different response from the same protein. It can go ahead and activate GSK3, which can help with the conversion of formation of glycogen, or it can block PepCK and 6 G6Pase and so on, which will block the glycogen formation pathway. So what we are seeing is that either the insulin receptor changes, or the phosphorylation of insulin substrate changes, loss of suppression of PepCK, and the translocation and activity of GLUT4. So all of these things can change. So the other way that signal transduction can be affected is by the use of chemical analogs or drugs. So in a natural situation, we have an ubiquitous receptor binding to a natural substance which acts as the ligand, leads to normal cellular activity. When we have an agonist drug, what it's going to do is it's going to go and bind in the place of the natural substance, and therefore it can activate the signal. So even when the natural signal is not there, because we are introducing a molecule that is very similar to the original molecule, it's going to create a enhanced cellular response, because there's many of the molecules that you are injected that are coming in or taken in that are coming in. Then we have the antagonist drug, which are basically going to act competitively. So your antagonist drug will come and bind to the antigen binding site and change the way that a natural substance is able to bind to it. So it's going to block it from binding. So those are, again, just to reinforce, two different ways, by mutations in the signal pathway, or mutations on the receptor protein, or mutations in the signal transduction pathway. That's all one. And then we have molecules that activate or inhibit them. So hopefully you met all of your learning objectives. We looked at cell communication, ligands and receptors, reception, transduction, and response as the three parts of a signaling response. And we saw that there are different types of cell communication: direct, local, long distance. Major type of receptors are GPCR, TRK, and ligand-gated ion channels, and kinases and signal cascades and amplification, varying responses and control. These are things you need to know about the signaling pathway. And finally, we saw the need for homeostasis and how negative feedback and positive feedback contributes to it. And finally, changes in signal transduction pathways. Okay, so hopefully this was helpful. Thank you very much.